Hi, and good afternoon, everyone. Probably we are Italy and South Africa, but maybe there is also somebody in other parts of the world. So good morning. And yes, I'm really pleased and I want to thank Lucille because we recently met in a workshop on One Health in South Africa, and she invited me for this session. So I'm very, very happy to share what I know about zoonotic pox viruses with you. So this is me, but Jeremy already said something. So I'm a vet, as he said, but I moved 3 years ago to the medical school. And this is because we would like to run some research on zoonotic agents. After the COVID-19, everybody really acknowledged the importance of an interdisciplinary collaboration. So this is the summary of my presentation today. So I will try to give you an overview of zoonotic pox viruses. I would like also to point out a very important aspect of these diseases, because these are really neglected zoonotic diseases. So I would like to try to explain how and why diseases become neglected. So I will do it through the brief history, which is not so brief for pox viruses. I will also let you know about the pox vaccine era and how this is linked to what we are now seeing about monkeypox and other pox viruses that are emerging and re-emerging. And I will try to discuss with you some future perspective. So when we talk about the family Poxviridae, this is a very huge family. We have two subfamilies. One is called Entomopox virinae, and it includes all the viruses that infect insects, and the subfamily Cordopox virinae, which include all the genus and species that infect chordates, so vertebrate animals. In particular, today I will try to let you know what I know about Parapox viruses and Orthopox viruses. You see in brackets, the prototype species for Parapox virus is Orthovirus, which is a virus that probably you all know that infects sheep and goats and human. While the prototype virus for Orthopox virus is Vaccinia virus. But as we know, Orthopox virus is a huge family. We will talk about this very, very soon. And variola virus, so the virus that was causing smallpox, so variola major and minor, that were eradicated thanks to vaccination, belong to this genus. I would like just very briefly to talk about the genome of these viruses, which is a linear DNA genome, double-stranded, and it's a huge genome for viruses, so it's more than 100,000 kb, and for avipox virus, this can reach 300. Why I'm talking about this? Because the genome of this virus is quite conserved in the organization. So as you can see in this slide, all the pox viruses have a similar organization of the genome. So you have a central part of the genome, which is the part that is dedicated to essential genes. While you have two variable regions, one in the left and one in the right, and there are located unessential genes. And these are the ones that encodes different virulence factors. And this is very important because for a long time, these genomes have been engineered. In fact, as you know, pox viruses are used as vectors for vaccination, but also vectors for gene therapy. And in fact, what we modify in this genome are in fact the two variable region on the left, on the right, so you can easily take off the virulence gene and put in different genes that can be used for protecting the patient for vaccination or to cure some genetic diseases. So I would like also just to briefly mention the morphology and structure, because as you can see, these viruses have a complex structure. They are quite big. So we barely see them at an optic microscope, but they are quite different. So Parapox viruses are smaller. ovoid, and they have this crisscross symmetry that is very easy to recognize at electron microscopy. So if you have a scab or a pathological sample, it is very easy to find these virions. Orthopox viruses are bigger, and they have this particular looking with this projection on the surface. This complex structure that can have or not the presence of envelope confers to these viruses a lot of resistance. So these viruses are really resistant in the environment. And this is another very important feature when it comes to control and prevention. These viruses have a quite complete genome, and in fact, they, despite the fact that they are DNA viruses, they don't need to go into the nucleus of the cell. They can replicate in the cytoplasm because they have their own polymerase, and they also have a lot of other genes that are also able to correct some error during the synthesis of the new DNA. And these viruses are able to go out of the cell in two different ways. So they can cause lysis. So in this case, you don't have the envelope, or in other cases, they use a kind of budding, and because they use the Golgi apparatus to go out of the cell. So in this case, they have the envelope. Normally, when we deal with viruses that have the envelope, we know that they are not so resistant in the environment. But in this case, envelope is just another membrane that can be on these virions, and it doesn't really affect the resistance. These viruses are really resistant, and this is why they are also used for vaccination for thermostable vaccine, because they don't need really refrigeration. As I said, it is very interesting to go through the history of these viruses. So we know from a very long time about smallpox. We know that smallpox caused a lot of outbreaks. We know that smallpox was able to change the history because we know that when outbreaks came, they were able to really kill a lot of sovereign and kings and their generation. So really, it was able to shape the history of humans. And this is Ramses V that probably died of smallpox. You see here a picture of Jenner. Everybody know about the vaccination, the first attempt to use an heterologous agent to immunize humans against smallpox. We know that this practice was really not very popular, especially for the Pope and for Catholic and Christian religion, not only because of the practice, but because Jenner was using something that was derived from animals to really immunize human. So there were a lot of advertisements saying that after immunization, you could become like a cow. But this is very interesting because the approach of this vaccination was really a One Health approach that is very in the history of humans. As we all know, the smallpox vaccination, to really eradicate this disease, started at the end of the 60s, and that was a real worldwide vaccination campaign. And this was so important because we know that in 1980, the World Health Organization declared zero smallpox. And that was the first time that a disease in humans was eradicated thanks to vaccination. Why I'm saying this? We all know these things, so we don't have really to repeat. But it's so important because we know that Jenner and the agent that was used for vaccination of human was in fact not smallpox virus, but it was another species we think that is Vaccinia virus. I don't want to spend too much time on this because there is a lot of discussion about the agent that was used to immunize the world population. But it is important to underline that that vaccination was using an heterologous agent. It means that orthopox viruses can be really cross-protective. And this is another very important aspect that I would like to underline before going through my discussion. So cross-protection is a key in our discussion. So after 1980s, because we didn't have any more cases of smallpox, and thanks to the cross-protection, many orthopox virus infection were not evident anymore. These viruses became very popular in biotechnology lab. So that in our conferences in every 2 years, we just spoke about vectors, recombinant vaccine, gene therapy, and nobody was really talking about the diseases that are caused by pox viruses. So thanks to this vaccination, the diseases where this important disease was eradicated, a lot of other diseases were prevented thanks to cross protection. And in time, these diseases became neglected. So these are some examples of the use of poxvirus vectors to cure and to treat some tumors and cancer, and also the use as vaccine. What happened at the end of the 20th century? Something happened. In fact, some cases of poxvirus became evident in India, Pakistan, and they were associated with animal cases in buffalo. So at the end of the 20th century, there was another, wave of poxvirus infections, and again we started to hear about them. So buffalo pox became, very, very popular, especially for pox virologists like me, but nobody was really caring about this disease because it was happening in India, in Pakistan, in the global south. So we were dealing with that, but it was just affecting poor people in the rural areas. So that was not so important. But as we all know, unfortunately, something happened in 2001. In 2001, we had the September 11. September 11 was an unfortunate living lab for pox viruses, because in this period we had the resurgence of some cases of pox virus in Brazil. These pox viruses were linked, in fact, to Vaccinia virus. And the infection was transmitted from cow to the milking personnel, young male in particular. And at the beginning, everybody was thinking about the Parapox virus, but in fact, it was a Vaccinia virus. So at the same time, in 2000, we had buffalo pox, Vaccinia, and September 11. As you all know, after September 11, the US government, the NATO, and the United Nations, they were really scared about a bioterroristic attack through smallpox. So a lot of research was done to try to prepare the human population for possible pox virus attack, a bioterroristic attack. So there was a lot of research that was dedicated to setting up new molecules, as for example cidofovir and other, and military personnel and medical doctors, especially in US, were again vaccinated, because we knew that the immunity that was given through the smallpox vaccination that was stopped in 1980 was probably fading, so the young people were not protected. So after September 11, new research started to deal with pox virus as a disease agent, to prepare antivirals, first of all, first of all, but also to immunize the at-risk population, so military, doctors, nurses, especially the one that went to Iraq, because as you remember, everybody was thinking about a possible attack from Iraqi government. So another important threat that was linked to September 11, as you remember, was anthrax. So anthrax was another bioterroristic agent that was really on the news after September 11. As you remember, there were some attacks in the US with the spore of Bacillus anthracis. And the CDC was working on this to really try to detect all the possible cases of anthrax in humans. And so cases like this one were considered for possible bioterroristic attack with Bacillus anthracis. But what was evidence? It was that, as we know, Bacillus anthracis can cause respiratory and very severe form, but in many cases, it just causes skin diseases. So this scabs, this appearance was linked to Bacillus anthracis, but in many cases, these were in fact Parapox viruses. So this was Lederman and co-author published in 27 and before some cases that were linked to suspected case of Bacillus anthracis linked to bioterroristic attack that were in fact caused by endemic Parapox virus infection. So if you want to see, there is also some photo quiz like anthrax or HORF that were published during the time, just to keep attention, because the appearance of these diseases can be very similar. So I have mentioned Parapox virus and ORF. What we know about ORF? ORF is in humans is a self-limiting disease that is frequently linked to professional exposure. So it is quite frequent in farmers, in shearers, in butchers, and also in vets, of course. Normally, as I said, is self-limiting, but in some cases, the lesions are really serious, and they can cause high level of dailies and invalidating disease. So if you are interested, this is one of the most read review that is available on ORF. And what we think about ORF is, well, ORF is really widespread. But because normally it causes a self-limiting disease, many people that know that probably their animals were infected, so they got infected from a direct or indirect contact with infected animals, they don't really seek medical attention. And in our experience, I'm showing you a clinical case. This was a colleague of me, a vet. He had some infected sheep in his flock, and he got infected with ORF, and he went to the hospital because he had a very severe form with lymphangitis. You see here, this is a lymph node. So he sought medical advice in a hospital near Bologna. And he went there, but you know, doctors were quite sure that was Bartonella and it was not a viral infection. So they started an antibiotic therapy, and they didn't want to really consider the possibility that sheep was transmitting a disease to this vet. But then he called us and he asked just to do a PCR, and in fact it was ORF, and then we shared with our colleagues in Rimini Hospital that this was an ORF virus infection in fact, and so the vet stopped the antibiotic therapy, and more or less in 2 months he recovered. So, as I said, it's not so common, and especially outside of rural areas, medical doctors are not used to this kind of disease. This is why sometimes it is so tricky to have a proper diagnosis. Human-to-human transmission is not very common, but this is, for example, sorry, there is some Italian there, I'm sorry. This is a case of transmission between a mother to his son. And so it is possible, even if it's not so frequent. So normally humans get infected through direct or indirect, and I want to underline indirect because I said that these viruses are so resistant in the environment. So it is possible to just get infected through the contact of contaminated environment without really being in touch with animals. But in this case here is quite evident that the mother that was a farmer got infected from her animals and then transmitted the disease to her son. I'm showing you this publication because we did it in South Africa. With my colleague Sharon McCrindle, and we did an investigation throughout South Africa, and it appeared that ORF is really widespread. And 2 weeks ago, I was in the field in South Africa, and it seems that farmers are well aware of ORF in sheep and goats. But the link, well, from what I could really hear from them, so through a questionnaire or through some question to the farmer, it was not so clear that they know that the disease in humans can be linked to the manipulation of their animals or probably the contact with contaminated environment. What you can see in this map is that ORF is really widespread, and the only human case that we were able to detect was in fact a vet, and she knew about the disease, so and she got infected through the manipulation of infected goats. Okay, but ORF is not just causing very typical self-limiting diseases. This is a big lesion, a recurrent lesion in a patient with immune compromised. He had a failure of cell immune response, and he was treated surgically many times. So this mass was taken off, but in a couple of days, this mass was really there again. And as you can see, it was 1990, and unfortunately, this patient was amputated at the end. So only after the amputation, it was clear that it was not a tumor, it was not cancer, but it was ORF, a complicated case of ORF. Complicated case of Orthovirus can be seen also in immune competent. In this case, this is a farmer. He had a burning in his face due to an accident during his work, and he got infected through contaminated environment. So as you can see here, you can see a very atypical form of ORF in this patient. Indeed, also in animals, you can have complicated forms of the disease. Normally, sheep and goats are affected with lesion in the mouth, in the udder, but sometimes we have also generalized infection with different parts of the body that are affected. Thanks to our global collaboration on poxviruses, we've been in touch with some colleagues from the US, and we helped them in trying to sort out what was this kind of disease in human patients. And these are some secondary complications of Orthovirus in patients with a reaction, a self-immune reaction that was caused by a previous infection with Parapox virus. So even if the presence of the virus in this scab was really low, the virus was still there. So probably this patient developed a kind of very mild viremia. And after, sometimes from the first lesion, these immunobullous diseases appeared in the arms, in the hands, as you can see, in the trunk, in the mouth, and so on. So sometimes these parapox viruses cause also complications that may arise after a while from the first infection. Another interesting case is this one. This is a Moroccan patient. We helped some colleagues in Alessandria Hospital, and he was slaughtering animals with bare hands. You know, religious slaughtering is quite popular and is becoming popular in Europe due to the high presence of migration from Northern Africa and Arabic Peninsula. And this patient normally get infected during the slaughtering practice. You know, bare hands it's quite at risk procedure. We don't want to talk about welfare, animal welfare in this case, but we know that we have to cope with it. And this is another complicated form of ORF. So this patient had a lesion on his thumb here, and after several months, he developed this erythema multiforme on his hands and his arms. And this is another case of complicated form. Again, this was a farmer. He was from North Africa again, and he was in touch with infected cattle that probably had pseudo Cowpox virus, which is another species in the genus Parapox virus. And in this case, it was in fact bovine papillary stomatitis. So we know that bovine can get two different diseases, very similar in their appearance. One is caused by pseudo Cowpox, and the other one is caused by bovine papillary stomatitis. And in fact, this patient was positive for BPSV. Again, this is another case from Bologna. This patient, she was a woman, 70 years old, and she had a very big lesion on her thumb, and nobody really thought about the possibility that this was caused by a virus. So she underwent different treatment with antibiotics, with surgery, but unfortunately she never recovered. She was slightly positive for TB, but except for that, she was immune competent and she had no problems. But thanks to the anamnestic history of this patient, we started to collaborate with the human doctors, and we find out that she was a farmer. She went back to Albania, that was her home country, and she manipulated animals, preparing food. So probably she slaughtered some animals, or she was in touch with some raw meat that was infected, and she developed this disease. So the manipulation of animals can be a very at-risk practice. Of course, we know that many cases of these diseases are linked to a professional exposure. So farmers again, but also slaughters, butchers, shearers, vets can be easily affected. The problem is when you have these diseases associated with patients with no history of contact with animals. And this may happen because, again, these viruses are so resistant in the environment. And the CDC, for example, underlined the risk, for example, for kids that visit didactic farms or some urban zoo where you can have also domestic animals, and maybe they get in touch with domestic animals that are directly with lesion or contaminated environment. So sometimes the anamnestic history of the patient is not really helping, but it is very important to keep these diseases in mind when we have to deal with differential diagnosis. This is another case that we have seen in Bologna. And this patient, she was 43 years old. She was a farmer again. She developed this bullous lesion with scabs, and she was suffering with this recurrent lesion. So sometimes she looked like recovered, but then after a while she again developed a lot of vesicular lesion on the hands, on the arms. And thanks to her history, we realized that she was bottle feeding a calf, and she was bitten by this calf. And calves are frequently affected by bovine papular stomatitis. And I'm showing you some cases here. You see some lesion on the muzzle and in the mouth. So it is also this disease, there are not so many cases published. So sometimes when you do differential diagnosis, it's easier to think about ORF, but sometimes people are not in touch with sheep and goats. But it is important not to exclude other parapox viruses, because even if this there is not so many scientific evidence, because these are really neglected diseases, it is very important to keep them in mind when we have to do a differential diagnosis, so also to define a proper treatment. So thanks to these two cases that I just mentioned, so the woman with the big lesion on the thumb from Albania and this woman here, we decided to do a retrospective study on some cases that were stored in our labs. And many of them were due to suspected cases of skin leishmaniasis. As we know, leishmania is another neglected zoonotic disease. So we would like to see if these retrospective cases can be in fact caused by other agents, other neglected diseases, because 50% of these leishmania suspected cases were in fact not diagnosed. So there were no identification of any agent as responsible for the lesion. So we started from these more than 200 formalin fixed samples, and we performed different analyses to identify cutaneous anthrax and also Parapox virus. We were not able to identify any skin anthrax, but in fact we were able to identify different cases of bovine papular stomatitis virus and also some cases of Orthovirus. So it is very important to underline this result, because around 60% of the samples that were firstly suspected to be skin leishmaniasis, in fact, were Parapox viruses. So then we are publishing these results, and we performed a research on PubMed database between 2000 and 2022, and we have realized that more than 12,000 publications can be found for leishmaniasis, more than 4,000 for anthrax, while overall for orthopox virus and parapox virus in humans, there are less than 300. So we can really state that poxvirus zoonoses are twice neglected. First of all, because there is no official surveillance plan. They are not included in any list, either World Health Organization and OIE, unlike leishmaniasis and anthrax, which are at least in the list of OIE, and leishmaniasis is also in the list of WHO. And and in particular, there is a really small number of research groups that deal with this. So there are not so many publications that provide evidence of the spread of these diseases. So we concluded that these endemic viral diseases are widespread, and unfortunately, due to the fact that standardized protocols are not available for these diseases, they are not included in the routine differential diagnosis, but it's so important to establish a One Health approach, or at least an interdisciplinary approach to these diseases, especially when there is a history of contact with animal, to try to really identify the proper etiologic agent. So now I skip to the orthopox, because I think everybody is really waiting for monkeypox. I have already mentioned the pox vaccine era, and I have already showed you this picture, even if smaller, in my previous slide. So I said that at the end of the 20th century, we started again to see some orthopox virus infection in humans. I mentioned buffalo pox in Pakistan and India, and many cases of vaccinia virus infection in milkers in Minas Gerais and in other provinces in Brazil. What happened there? First of all, we knew that all the patients that were infected were not previously vaccinated against smallpox. So these people were not immunized, so they couldn't really count on a cross protection from the vaccine. The lesions were also present in the animals, so there was, in fact, the demonstration that it was a zoonotic transmission. But our colleagues in Brazil and the CDC started an epidemiological surveillance to really sort out what was the reservoir, because vaccinia virus and the sequence of this virus were so similar to the vaccine strain. So everybody was really thinking about what happened. So there is the vaccine strain that is circulating in the field. And in fact, what was shown, thanks to epidemiological investigation, there were a lot of rodents positive in around the rural areas where these human cases were detected. And in fact, small rodents like bank voles were found to be not only seropositive, but also positive for the virus without showing any clinical lesion. And this is very important because rodents are frequently the reservoir of pox viruses, but they very rarely show any lesion. So you have these animals that are perfectly healthy, but they are able to spread through droplets or through their feces the virus in the environment. And I want to repeat that these viruses are very resistant into the environment. So these are some publications, and of course, the first suspect for our Brazilian colleague was ORF, or at least a Parapox virus, because we know that Parapox virus in cattle are very common. The electron microscopy was in fact able to detect the morphology of orthopox virus. As you remember, I showed you at the beginning. So it was not a parapox virus, but it was a orthopox. And at the end, as I said, the genome was very, very close in terms of identity to vaccinia virus. And this is buffalo pox again. In Anthropocene, what is happening? Is happening that these diseases are really coming into urban settings. So what happened is that at the end of the 20th century, this lesion, this disease was really apparent in rural setting, in the global south. So nobody was really caring about. So they were really neglected diseases. But as you all know, in urban setting, there is a trend of having companion animals. Sometimes there are traditional companion animals, such as cats, dogs, and little fish, or others. But then we know also that in urban settings, exotic animals are becoming very popular. I would like to show you this slide just to give you an idea of the trade of wildlife in the world. So more than 5,000 vertebrate species are traded. Many of them are endangered species. And this is very important to keep in mind, because the trade of wildlife is just in the is ranking second place after drug for the amount of money that is bringing. So we have to keep in mind that people are so happy to have in their house a reptile, an exotic bird, amphibian, mammals, rodents, and so on. So we are treating what we previously considered pests as pets. And what does it mean in terms of diseases that can be transmitted to humans? Okay, so at the beginning of the 21st century, something happened again. And as you can see in this patient, this lesion in the first stage were suspected to be anthrax. But thanks to electron microscopy, it was evident that it was not a Parapox virus, but it was a Orthovirus virus. And after sequencing, it was very evident that these infections were in fact Cowpox virus. What is a Cowpox virus? Everybody from the name thinks that Cowpox virus is a poxvirus of the cow. But it is not the case. Probably in the past, Cowpox virus was causing, the variola in cow, and probably that was the first strain that Jenner was using to vaccinate people against smallpox. But in fact, we don't see so much cases. Well, we don't see any case in cattle. But we see a lot of cases of Cowpox virus in different wildlife species like elephant, rhinos, lions, leopards, and also a lot of domestic and peridomestic animals. This kid here was bitten by a fancy rat, and this fancy rat was completely healthy. So no clinical sign, but it was positive. It was really spreading cowpox with his droplets. So cowpox is causing diseases in many mammal species, but the reservoir again is in rodents. This lesion here is an owner that was bitten by his cat that has Cowpox virus. Again, cats can become infected because especially the cats that go out, they want to eat rodents and rats, and sometimes they get infected from this practice. And this is another important thing that I want to underline, because when we will talk about monkeypox, this is something that we will repeat. So it is interesting because poxviruses can be also transmitted indirectly through a domestic animal, such as a cat, and this may happen also for other viruses. So our domestic animals go out, they can be in touch with animals that are not really companion animals, but they can come home and infect their owner. This is another big lesion in a patient that was petting his mongoose, because sometimes mongoose are kept as a pet animal. In this case, the mongoose showed a clinical lesion, and she was in fact positive and transmitted the disease to her owner. So in 2009, the European Commission sent this informative. There were a lot of cowpox in human in France and in Germany, and they were all connected with the contact with pet rats from the Czech Republic. Now in urban settings, having a pet rat, what we call a fancy rat, is becoming very popular, but we don't have to forget that sometimes people that have reptiles in their home, they feed the reptiles with rats, even if they are dead but infected. There can be another way of transmitting the disease to humans. So this is a publication from an Italian group that showed a clinical case in a vet that was infected through the contact of a cat. So re-emerging zoonotic orthopox viruses are really associated with ecological changes, but also changes in habits of having companion animals, popularity of exotic animals, fancy rats, wildlife that can be used for food. And this is another very important aspect, because people are moving, we are in a global world, and people that can be in touch with us can have very different eating habits compared to the one that we are used to. So wildlife and bushmeat is not common in Europe. I don't know very well about South Africa, about the practice of having bushmeat. I know that you have this kudu meat that is very, very nice, but I don't think you eat other like monkeys or something like that, but you will tell me about that. As I said, reptiles can be another reason why people can be in touch with infected reservoir, and also zoo animal. So, and last but not least, orthopox virus cross protection is decreasing in the world population, because we stopped vaccinating against smallpox in 1980s. So we have to take into consideration that a large part of the population that will increase in the near future will not have any protection thanks to that vaccination. So it is quite clear that in the last 20 years, a number of cases were reported, and we expect that more will be reported. So I would like to finish with monkeypox. So these are the cases that were observed in the US in 2003. This is a kid, and this kid was in touch with an exotic animal, which is a prairie dog. And this prairie dog was sold in a shop, in a pet shop in Chicago. And the prairie dog were not the first cause of monkeypox infection. In fact, prairie dogs became infected through the close contact with other rodents that were coming from Africa. And this is this guy here, a big one, a big rat that is called Gambian Rat. So in this pet shop in Chicago, there were some African rats in close contact with local reproduced rodents in America. And they were the origin of a cluster of around 50 cases of monkeypox, especially in kids. Many of them resolved with just a skin lesion, but some of them were really serious with also neurological symptoms. We have to take into consideration that compared to Parapox virus, orthopox virus are systemic diseases. So they can cause viremia, fever, and also an enlargement of the lymph nodes with a flu-like syndrome. So yes, this was the origin of the outbreak in the US. So after 2003, we started to talk again about monkeypox. We know that monkeypox is the closest relative to smallpox. This disease is widespread in Africa, especially sub-Saharan Africa, Congo, and many other countries. It causes a systemic disease, and the disease is thought not to be transmitted in a very fruitful way from and among humans. It is thought that the practice of consuming bushmeat can be the main risk factor, even if the name, as we all know, comes from the discovery of monkeypox in laboratory monkeys. In fact, also in this case, the rodents are the reservoir. So monkeys become infected through the contact with rodents in the field. This is a video. I don't want to take too much time just in case I will share my slide, but this is telling us about the outbreak of monkeypox in Singapore just a few years ago. And this was really linked to a guy coming from Nigeria that was interviewed, and he declared to consume bushmeat in his home country. So, sorry. Want to show you, okay? So where has monkeypox been detected until 1 month ago? We knew that a lot of cases are happening in sub-Saharan Africa, and these are all the countries where they were really detected. Some of them are endemic countries, or are thought to be endemic country. Some other have been identified as country where some imported cases have been detected, but we think that this disease is really widespread in all sub-Saharan Africa. US is the outbreak that I was telling you about in 2003. After 2003, there was a ban in importing any exotic animal in the US. So if you would like to have an exotic rat, it should be reproduced and farmed in the US. So you cannot really bring any animal from Africa or other countries anymore. Some cases were detected in UK before these latter ones that we all know, some cases in Israel, and I mentioned also the cases in Singapore. So these were, you know, the first alarm bells for everybody that something was changing. So we all knew that monkeypox could become an emergency due to the fact that people are really traveling for work, for holidays, and there are a lot of movement from sub-Saharan Africa toward Europe and the US due to migration, the practice of bushmeat, the practice of having exotic or importing exotic animal. So we knew in the field of poxviruses that this could happen. And this is these are the last cases that are reported by ECDC on the 25th of May, and these are the cases that are reported in Europe. As you know, the World Health Organization identified some cluster linked to some prides in Spain. In fact, a lot of cases were detected in Spain and Portugal, but we think that probably there are other reasons. We don't know so much about eating habits or possible contact with animals, so this information are not available at present. These lesions here are pictures taken from a patient that was hospitalized in Bologna. As you can see, the lesions are very few. In fact, this can be classified as a mild infection. And it seems that this case was in touch with another male having the same lesion. So it was a real man-to-man transmission. This is very important because we are learning at present what monkeypox can look like outside Africa. So we think that probably animals are more ideal as reservoir. They probably spread higher viral load. And so the lesion that we normally experience in Africa in humans are the ones that are really linked to a contact, direct contact or indirect contact with animals. While the lesion that we are observing in local cases are like this, so quite mild. And this is 1 week after the onset or the hospitalization. So you see that at the beginning is a blister and then a scab is forming. And this patient declared to have some fever just for 1 or 2 days and lymph nodes that were enlarged, but despite this, he was quite well. Very briefly, many other orthopox were detected after 2000. And so this is just to let you know that there are some poxviruses that are not really included in the taxonomy at present, because there are some researches ongoing on their genome. So some of them were detected from direct contact with animals, some other were associated with immune suppressed patients, which are the ones that suffer the most. And this is another pox that is probably another parapox case in the United States just a few years ago. So to conclude, I would like to give you some take-home messages, and then I will be very happy to answer your question, if any. First of all, it is crucial to maintain reliable information and possibly international prevalence data and detailed case histories for infection and incidence. Many times neglected diseases are neglected because nobody is doing research, nobody is doing surveillance, but this doesn't mean that these diseases are not important and are not affecting the health and welfare of people, and sometimes also animals, and in these cases, as I have mentioned. It is very important and crucial to establish a cross-sectorial collaboration, at least between vets and meds, to deal with zoonotic diseases, because everybody is taking care about health and welfare of animals, and we have to consider that humans are mammals, are animals, and viruses cannot really see the difference between a human or a cow or a rat. And I think zoonotic diseases tell us a lot more about One Health than what we can learn from books or from lectures. It is very crucial to adopt a One Health perspective, not only because we need to exchange between the veterinary and medical sector when we deal with zoonotic diseases, but we have to consider that biodiversity, environment, climate change, and many social factors like migration are so important, or cultural practices. I mentioned religious slaughtering, bushmeat, and many other that can be so important. So when we deal with One Health, we really need to open our mind and try to collect as many information as possible from very different disciplines to have a clear picture of what is happening in the field, because we really deal with complex system, and it is not anymore possible to adopt a linear approach to zoonotic diseases. And this is not only important to prevent the impact on human and animal health and welfare, but also to reduce the use of antibiotics and antimicrobials when they are not really necessary. And we know that AMR is so crucial, and it is the so-called silent pandemic. So having an etiological diagnosis is so important also to undertake a proper therapy. Thank you very much.
Wow, thank you so much. Hugely appreciative. We've got lots of great comments and appreciation in the chat there. Alessandra, I'm going to just go through some of the questions because there were lots, and that's a really good sign. One of the questions was just to check about the transmission. I know it may differ a little bit by particular viruses, but does it have to go through in a broken skin or mucous membrane, or can it penetrate through intact skin?
Well, when we talk about Parapox virus, for example, it is well known that you need to have a lesion. So any lesion. Of course, farmers are the ones that do not have the hands of a medical doctor, or sometimes they really have very hard hands, and they may have just micro lesions. So these viruses really need to go into the skin through a lesion, while orthopox is very different because they use droplets. So they really don't need to have, well, you can also have a transmission due to a lesion in a mucosa or in the skin, but the most relevant way of spreading these viruses is through aerogenic transmission. And this is quite similar to what we experienced with COVID, even if it's a totally different virus. So masks can be useful also for this.
Yes, agreed. One of our colleagues is asking, are any of the pox viruses oncogenic in terms of causing cancer? Because some of them look quite tumor-like.
Yes. Yes, the one that I mentioned are not really tumorigenic, but there are some pox viruses. I didn't mention other, like Yaba pox or Tanapox or others that are really causing tumorigenic transformation of tissue. These are very tricky viruses, very interesting to study. But yes, not the ones that we have mentioned. So the lesion that you have seen are in fact caused by a failure of the immune response of the patient. Because all of them encode virulence factor. One of them is VEGF, that cause proliferation of the cells and also the endothelium. And so VEGF is probably one of the main factor that is really on the basis of this giant lesion that we have seen.
Yeah, that's a good point. Another question that's come through is about vaccines for animals for the pox viruses. Presumably they do work well? I mean, or is it only against some?
Yeah, regarding Parapox virus, they are not very effective.
Okay.
Because these viruses are very tricky, very interesting. In their terminal region, they encode a lot of immunomodulatory factors that are really playing with the immune system. With normally, when you have a vaccination with a traditional vaccine, first of all, they produce anyway lesions. So you have scabs after vaccination, and these scabs bring live virus in the environment. And the other one is that after 6 months, the animals are again susceptible to the disease. Regarding other pox, we have very good results with, for example, sheep and goat pox and also lumpy skin disease. They work very well. And so probably we can do it also for orthopox, but because we were so happy that we didn't have orthopox anymore, thanks to smallpox, we didn't do any research.
That's a good point. And this follows on, you did mention some of this just right now, but another person's asking about how long immunity is conferred after these infections. So Parapox, not very well. Orthopox?
Not very well. Orthopox, we know that it's a long-lasting one. It's a long-lasting one. We are not sure about, for example, I was vaccinated. I'm not so young, so I was vaccinated, but nobody took me blood to say, you still have some antibodies. There was a wide discussion about that during one of the pox meetings that we had. We are unsure about that, but the fact that many young people, the ones that were not vaccinated, are more affected, we think that probably some memory in our immune response is still there for the ones that were vaccinated with smallpox.
Yeah.
Yes, smallpox.
Yes, agreed. And then, of course, the question that we always worry about in South Africa with such a high burden of HIV and AIDS is about the cell-mediated immunity with some of these, particularly Parapox, I think, but both, right? Orthopox and Parapox.
Yes,
it's kind of key.
Yeah, in fact, what we think is that probably in HIV patient, you see this very atypical lesion. So it is not so common that a medical doctor wants to detect a virus there. Maybe they think that it's just a tumor or something like that. So it is very important to take this into consideration, especially in patients that live in rural areas, because atypical lesion in immune suppressed patients are very common.
Yeah. We'll cross our fingers and hope it doesn't become a big problem here. And then a last question, I think, everyone is, you must read the chat afterwards because people are very, very grateful for your conversation. But one final question, if you have the time, just asking about the smallpox vaccines with the recent reemergence of monkeypox. what I think for me at least, to me it appears early days. We don't know how big this outbreak's going to get, but if it doesn't, and if we get lucky and it dies down in the next few months or so, it does sound a bit like a warning shot, because when the world needs this, we suddenly find out we don't have enough smallpox vaccine to give to low and middle income countries particularly. I don't know if you had any specific thoughts on that.
Well, of course, what we expect is that we will see more orthopox viruses infection in the near future, because this cross protection is really fading in the world population. We don't know if the World Health Organization will decide to go with vaccination again, because with COVID, we experienced that people are not so happy to be vaccinated everywhere. And the situation is very different from what we had in the 60s of the 20th century, where I remember we went to school and we were vaccinated. So our mother couldn't complain about our vaccination. We had to be vaccinated because we couldn't access school if not. So yeah, we have to take into consideration that the world population has changed their mind in vaccine, and even with COVID was so a severe disease, people decided not to vaccinate. So we will see what happens. Of course, we have vaccine that works very well. So.
And sorry, I'm going to sneak in one last question, if that's okay, just about the possibility of a pan-pox virus PCR. Because at the moment we do individual pox viruses, aren't we? Yeah.
There are a lot of protocols available online. If you just go to PubMed, you will find a lot of panpox and pan-orthopox, pan-parapox. So you can have real time, you have LAMP, you have a lot of means to detect these viruses. So why we don't perform any surveillance?
Yes.
So easy.
Agreed. Well, thank you. I think you've changed a lot of people's minds about that today. So thank you very much for your time. And we're really appreciative. And like I said, please, before you log off, have a look at the chat. There's lots of messages thanking you for a wonderful talk, and I can only agree. So thank you very much for your time.
Thank you for the invitation, and I hope to be in touch with you.
Sure, of course. Thank you.